So now let's see how the transcription actually occurs. For the transcription to occur, the first and the foremost thing is formation of pre-initiation complex. See, we have already seen DNA is in the form of a double-stranded molecule. So this is one strand of DNA, that is the template strand, and here is the other strand of the DNA molecule. For the formation of pre-initiation complex, we need many transcription factors. These transcription factors actually helps in recruitment of the RNA polymerase enzyme. So, first and foremost, transcription factor 2D comes. This 2D transcription factor has got one subunit or TBP, that is Tata binding protein, as its subunit. So this Tata binding protein comes and binds to the template DNA and it actually bends the DNA molecule. So this transcription factor 2D comes so that Tata binding protein binds to the Tata box in the promoter region and it helps in the bending of the DNA double strand. After that, either transcription factor B comes and after that transcription factor 2A can come, or it can be the other way around. First transcription factor 2A can come, then the transcription factor 2B can come. So after these three joins, then transcription factor 2F comes and binds to these three. It will bind to transcription factor 2D, transcription factor 2B, and also, which is not shown here, it will bind to transcription factor 2A also. So what is the importance of transcription factor 2F? Its main function is recruitment of RNA polymerase enzyme. So transcription factor 2F brings the RNA polymerase enzyme to the template site at the promoter region. After that, transcription factor 2E comes and in last transcription factor 2H comes. They all come and binds to the DNA molecule at the promoter region. So what is the main function of transcription factor 2B? As we have discussed earlier also, transcription factor 2B interacts with the core promoter element BRE, that is B recognition element. And transcription factor 2H, it has got mainly two subunits, alpha and beta. Its main function is, that is transcription factor 2H, it has got two functions. One, it has got the helicase activity, that is it melts promoter region to form the open complex, that it unwinds the promoter region to form the open complex from the closed complex. The other is its kinase activity. This kinase activity phosphorylates the serine moieties in the CTD. What is CTD? This is our RNA polymerase 2 enzyme. It has got the C-terminal region here at the tail. So it's the C-terminal domain of RNA polymerase enzyme. See, all the enzymes are proteins. So in the tail region, it has got many serine amino acids. This kinase activity phosphorylates the serine in C-terminal domain of RNA polymerase enzyme and it helps in the promoter clearance. What is promoter clearance? When RNA polymerase enzymes clears the promoter region and moves towards the structure region, that process is called promoter clearance. So kinase activity phosphorylates the serine residues in the CTD and helps in the promoter clearance. So for RNA transcription, we need the enzyme RNA polymerase. Enzyme RNA polymerase needs magnesium ions both in prokaryotes and eukaryotes. In prokaryotes, it is made up of two parts. One is the sigma, the other is the core enzyme. When this core enzyme binds with sigma, then you get the holo enzyme, that is the whole functional enzyme. Sigma part, it's abbreviated like this, it actually helps in the recognition of consensus sequence in the promoter region. So, what are the consensus sequences? These are the frequently and constantly occurring sequences in any molecule. So, in the case of prokaryotes, we have got the Pribnobox or TT-GACA or some other sequences like this. This sigma molecule actually recognizes the consensus sequences in the promoter region. So, it actually helps in the binding of the holo enzyme to the promoter region, and it provides specificity to the holo enzyme. That is, it binds with some specific consensus sequences so that only those genes are activated which have got the specific consensus sequences to the sigma molecule. So, it provides the promoter specificity. What are the other part? Other part is the core enzyme. Core enzyme has got many subparts, like it has got the 2 alpha, it has got the 2 alpha regions, then beta region, then beta 1, and then the omega. This alpha region actually helps in the binding to the promoter region. Beta is actually, it acts as a ring around the DNA molecule or around the template strand, so that this enzyme does not wanders away from the template strand and keeps on making the RNA molecules. Then beta 2 actually has got the 5' to 3' polymerase activity. This is the functional enzymatic part, this beta 1. It has got the polymerase activity, but it can only binds new nucleotides from 5' to 3' direction only. Omega is the smallest subunit and it stabilizes the core enzyme. So, omega is for stability. Beta 1 is the actual enzymatic part which binds the new nucleotides from 5' to 3' direction. Beta 2 actually makes a ring around the template strand, and alpha units bind to the promoter region. But the promoter specificity is provided by the sigma subunit. Then when the capital E, that is the core enzyme, binds with the sigma subunit, you get the holo enzyme. So what is 5' to 3' polymerase activity? Suppose this is one nucleotide, this is our sugar moiety, this is the nitrogenous base, and this is the phosphorus moiety. This is the 3' end of the sugar moiety, this one, this is the 3' end, this is the 5' end. So when new nucleotides come, this enzyme polymerase can actually bind the new nucleotides like this. This 5' can bind to this 3', so that the ultimate this dimer, its direction will be from 5' to 3'. See, this is 5', this is 3', then again the 5' comes and the 3' is left. So, new moieties always binds at the 5' region only at phosphorus molecule. So that the ultimate molecule or the chain, polymeric chain, it will get the direction from 5' to 3' only. On the head region, 5' will be free, and at the tail region, 3' will be free. So new molecules are always attached at the head region only, that is at 5' region. This is known as the 5' to 3' polymerase activity. It is never the other way around. It is always from 5' to 3' region only. So in the case of eukaryotes, the polymerase again will need the magnesium enzyme. If we see the structure of the polymerase, then it has got two large subunits that will be similar to beta and the beta prime. See, beta prime is the actual active subunit with the 5' to 3' polymerase activity, and beta is needed so that it forms a ring around the template strand. So in case of eukaryotes, we will have two large subunits similar to beta and beta dash plus around 14 smaller subunits for various function. Then in case of eukaryotes, more than one RNA polymerase can transcribe the same template strand simultaneously but at different sites. See, in the case of eukaryotes, this is one chain, this is the other strand of DNA molecule, this is the RNA polymerase, and it will keep on transcribing the RNA molecule like this. But in case of eukaryotes, so it will keep on transcribing one RNA molecule like this. Suppose this is the enzyme and this is the RNA chain that is being made. But in case of eukaryotes, there will be many RNA polymerases at different sites. So this RNA polymerase is making this chain, this RNA polymerase is making this chain, and this RNA polymerase is making this RNA molecule. So there will be many RNA polymerases that are transcribing this DNA template strand at different sites simultaneously, so that we'll get the many new molecules of RNA molecules in a shorter time.